Hi, Mark. Would you like to start by introducing yourself, Mark? Let us know what you do.
Of course. Okay, so my name is Marc Hummersone. I'm the Senior Director of R&D at Astrea. And, to give you a brief potted history of who I am, I guess, a good place to start is the beginning. So I'm a qualified chemist, and I've been involved in biotech since finishing my postdoc in the total synthesis of natural products. I initially applied my skills to drug discovery and development, and was proud to be part of the team that brought the PARP inhibitor Lynparza to market. And it was around about that time that I started to learn and hear more about biological therapeutics. And I developed an interest in how they're produced, and it quickly became apparent that there was a clear and unmet need for purification solutions. I then had the opportunity to become part of a company that was developing fiber-based chromatography technology and became the chief scientific officer. That company was called Purify, and we worked away before being acquired by General Electric Healthcare. GE Healthcare then became Cytiva, where I was the director of fiber chromatography at Cytiva. More recently, as I mentioned, I've had the opportunity to join the Astrea team as Senior Director of R&D. And my role here is to oversee the R&D function and to contribute to the business strategy while developing innovative new products for Astrea and customers alike. And I work with a fantastic team who, like me, are dedicated to delivering to the needs of the customer and the patients. And in this regard, I've got a particular interest in serving the needs of the cell and gene therapy sector, which definitely requires next generation purification solutions. So in a nutshell, that's me.
Brilliant. Yeah, so you've been in this industry for a good while, and you've seen all the changes, I'm sure, that have been happening throughout the years. Would you like to comment on how the industry has evolved over this time?
Sure. So I think for me, some of the biggest changes that have been observed are around the manufacturing efficiency, and this has allowed the industry to address what was once a bottleneck for manufacture. And that was seen several years ago, but now that this has been overcome, it's a really good thing to see, and this has been mainly attributed to expression systems that have allowed the industry to produce more grams per litre of target molecule without having to increase the size or number of bioprocessors. And this in turn has been driven by improvement in cell lines and growth media, and that's helped to push the increase in titers forward too. And in addition to that, I see the adoption of single-use components in manufacturing, and that's made a real impact where producers are able to use single components to avoid laborious cleaning and quality control of reusable stainless steel devices. The increase in productivity from the upstream guys has really required innovation in downstream bioprocessing. And this has required the downstream processing guys to come up with solutions such as high throughput devices for effective bioprocess optimization, and use of disposable systems, continuous upstream processing, continuous chromatography, and integrated continuous bioprocessing, among several other techniques as well. But I guess what I'm trying to stress here is that the pressures from the upstream productivity have forced innovation in downstream bioprocessing, and so that's really where the industry has changed significantly over the last 20 years.
That's amazing. So, is the downstream processing the key area for Astrea? Is that where your focus is in this cell and gene therapy sector?
Absolutely. So for us as a company, we are focused on downstream processing solutions. That's around, as I mentioned before, when I gave you a little bit about my background, explaining that we're focused in the area of cell and gene therapy, providing solutions that don't currently exist right now for cell and gene therapy. And we're all about downstream, downstream bioprocessing. Yes, 100%.
You're deep into the downstream processing part. What do you think needs to be addressed, and what are you guys focusing on? What technologies are you working on?
So, I think there have been some significant advances in the purification media, and that's around resins and membranes. And higher productivity upstream processes are driving higher capacities and speeds in downstream processing technologies. This is most clearly seen in things once upon a time protein A The highest binding capacities we could get were 20 megamil. Now we're clearly up to 100 megamil. So there have been improvements, but where do the further improvements need to be made? And I think that this relates, again, to the point that I made earlier, but it's all about throughput, and that throughput in combination with quality. And, we've spent so long using resins and focusing them on MAB products that we're trying to repurpose them for cell and gene therapy. And new strategies using new materials that are dedicated to cell and gene are the only way forward, not repurposing old tech. And to put that in perspective, if we consider within cell and gene, most of our targets are actually quite large compared to the regular monoclonal antibodies that we were trying to purify in the past. The pore sizes of resins aren't big enough to allow us to use resins effectively on those larger targets. So why are we still using resins for those new targets? At Astrea, we're working with our partner companies within the Gamma portfolio of companies, and most notably Nanoparel, to develop fiber-based chromatography media where the pore sizes are huge, and they work much better with these larger modalities that we're trying to purify in cell and gene therapy. So, overcoming the issues that old tech presents to us, rather than repurposing it, we should be developing new tech. That's where we're focused.
That sounds great. So in this new tech that you're developing, do you think it will address many of the common problems for, say, AAV purification for gene therapy, things like empty full capsid number, endotoxin level, and affordability? Do you think this will be able to solve a lot of those problems?
This is an area that Astrea have been focusing attention on, and we're looking at both single-step and multi-step purification in order to provide AAV capsids at as near to pure full form as possible. However, I don't think it should be restricted to just the downstream guys. So if we could incorporate strategies that could be useful, for instance, taking validated process analytical technology to measure the ratio, characterize the ratio during manufacturing, to find optimal fermentation, harvest, and processing parameters. This would be great. And if this is optimized, it would feed into resolving the full and empty capsids using established bioprocessing equipment to be more productive. And that would be using IEX and using mixed mode media. But because we've optimized our upstream, it would allow the downstream stuff to be even more efficient. We also have separate programs where we're looking at minimizing the processing steps. And that's specifically around reducing sample loss of all capsids during the purification process. So, we're looking at this in several different ways. You also mentioned, about making AAV-based gene therapies more affordable. The emerging tech that I see is focused around the needs to move from fit-for-purpose tools, as I mentioned earlier, to purpose-built ones for viral vector manufacturing and purification. Fortunately, companies like Astrea have been working both through internal R&D efforts and also in partnership with customers to develop new purpose-built products. Leveraging this experience is definitely going to be crucial in addressing the challenges presented by the urgency and uniqueness of gene therapies. Equally, affordability, that will definitely come with process compression. And here at Astrea, we're working with Nanoparel, and we're working on compressing the time and the resource it takes to purify AAV-based therapies through the use of cutting-edge ligands and high-throughput purification media, and that means using nanofiber materials. So using a combination of these cutting-edge ligands and new nanofiber materials, we're able to compress time, and therefore the cost savings are considerable.
Sounds like an absolute game changer. I'm really excited to see what's coming out of Astrea in the near future.
We think it is. That's why we're focusing on this generation of purification.
Yeah. So how has VectorBuilder helped you with developing these new technologies? How have we been useful to you?
That's a really good question. I'm glad you asked, because we have got experts here at developing bioprocessing media, but we can't do it unless we've got relevant feed streams to check on how our prototypes and how our designs are working. And so we work very, very closely with the guys at VectorBuilder, who give us the benefit of their expert advice and the expertise in the upstream side of things, to allow us to understand more about the feed stream so that we can develop purification media that is market relevant. And as such, they're our preferred partners in this sense, and it's absolutely a fantastic working relationship that we have with them, and one that is allowing us to move forward at pace on all of our projects.
Amazing. Love to hear it. Love to hear that we've been so useful to you. Yes, and looking forward to building on our partnership in the future, because I think we've got some big plans to test out your purification systems.
It would be great. And I think, like for the products that we're developing to be truly, beneficial to the industry, working with, people at the cutting edge like VectorBuilders is really important, because not only do you get the opportunity to evaluate a product, but the input in how to develop products that are industry relevant is really, really important.
Exactly. Thank you so much.
No problem. It's been an absolute pleasure speaking to you. I'm looking forward to working in more depth with the guys at VectorBuilder and yourself. And it's been an absolute pleasure talking to you. Thank you.
Absolute pleasure as well.